Processes in recombinant DNA technology. In this, we will study steps involved in construction of recombinant DNA and production of desirable product in large scale. We will study the basic steps. The steps involved in construction of recombinant DNA technology and production of desirable products or product are as follows. Isolation of DNA. Fragmentation of DNA by restriction endonucleases. Isolation of desired DNA fragment. Fourth step, ligation of DNA fragment into vector. Fifth point, preparation of competent host. Sixth point, insertion of recombinant DNA inside host. Seventh, large scale production. Eighth, and the final point, extraction of desirable product. First step, isolation of DNA. Firstly, the DNA must be isolated, means the desirable fragment, the desirable DNA fragment must be isolated from the source. Suppose, for example, I want large scale production of insulin. I hope you all know insulin is a hormone which controls blood glucose level. So if I want to produce if I want production of insulin in large scale, firstly I have to identify and isolate the gene or genes responsible for the production, the gene or genes which produces the genetic material. Now, how isolation is done? So, for DNA, we all know that a cell consists of several macromolecules. Just a moment. A cell is made up of different macromolecules, that is molecules which are in larger quantity, including carbohydrates, that is sugars, carbohydrates, proteins, nucleic acid, two types, nucleic acids, that is DNA and RNA, etc. These are the main types of, and also lipids or fats. So, to isolate DNA, all other macromolecules or the components present in a cell must be removed so that only DNA remains. This is done by two methods. One method is using chemicals and another method is using centrifugation. To isolate DNA, it must be separated from all other cellular components. This can be done by two methods. Number one, centrifugation, that is applying centrifugal force, or we can say rotated at a specific speed per minute, so that the the components of the cell get separated from each other according to its concentration or density. And next one is using enzymes. This we will discuss using enzymes. Now the enzymes used are as follows. So enzyme, just a moment, I'll write in the next page. Enzyme macromolecule removed. By breaking it down. So name of the enzyme, number 1, protease, breaks down protein. Then lipase, breaks down lipids. Then cellulase, breaks down cellulose, present in cell wall of plants. Then chitinase, breaks down chitin, present in fungal cell wall. And also another lysozyme, breaks down component of bacterial cell wall. So all these components are used. One more is there, that is ribonuclease. Ribonuclease breaks down RNA. So this only remains DNA, and the thread-like DNA is isolated in a test tube, as you can see in this image. Okay, so once RNA is removed, then ribonuclease, then purified DNA ultimately precipitates out after addition of chilled ethanol. So after applying all these elements, then purified ethanol is applied, which helps isolation of DNA in form of thread-like structure, which is removed by spooling. It is collected by spooling. Okay. So once, I repeat, once all of these enzymes are applied, after that the purified DNA precipitates, and when chilled ethanol is added. And the last step is isolated using spooling. Isolated using spooling. Now coming to the next step. As you can see, next step is fragmentation of DNA by restriction endonucleases. So next one, number 2, fragmentation of DNA by using specific restriction endonuclease. So as we know, restriction endonucleases are the enzymes which cut DNA in between 5' and 3' end at a specific location. This I discussed earlier, and you can see the recording also. So how a DNA is fragmented? specific enzyme specific enzyme must be used to cut both desirable DNA fragment as well as the DNA which will act as vector. This makes ligation, that is next step, easier. Okay, now coming to next step or next, third one, separation of desirable DNA fragment or isolation. First, we isolate the whole DNA from the source, from the cells. Then next, DNA is fragmented, broken down into several pieces using a specific restriction endonuclease. Same restriction endonuclease applied to cut the DNA carrying the desirable fragment as well as the vector in which the desirable fragment will be introduced. I repeat, same restriction endonuclease is used to cut both the DNA carrying desirable DNA fragment as well as the vector molecule. This makes ligation, that is joining, easier. Now coming to next step, that is isolation. Once fragmentation is done, then isolation of desirable DNA fragment must be done. For this, gel electrophoresis is used. Gel electrophoresis is used to isolate DNA fragments according to their size. DNA fragments according to their size. In gel electrophoresis, the gel used is called agarose gel. This is image showing gel electrophoresis. Just a moment. The gel used is agarose. It is a polymer of agar, which is a chemical obtained from seaweeds. Now coming to it is porous in nature, polymer of agar and porous in nature. It is because it is used, agarose is used in gel electrophoresis because it have pores and DNA can, when electricity is passed, DNA gets separated as it can travel through the pores. Okay, now so what else? Agarose is used due to its porosity. Now coming to the method. DNA is negatively charged due to presence of phosphate, that is PO4 minus due to presence of phosphate bonds. When electricity is passed, it tends to get separated. It tends to move towards cathode. Cathode according to its size due to porosity of the agarose gel. This is the method. This is used in isolation of desirable DNA fragment. In the next, I will discuss in this detail in the next part 2, isolation of desirable DNA fragment.